This is a supplementary slideshow for immunity taken from A Human Anatomy and Physiology. It happens to be the 6th edition by Elaine Mariab. Um, so I've edited this down to the bits that were left out of the other presentations. Okay, so we did talk about immunological memory and the primary response versus the secondary response. So during the primary response, I did make mention of this. This first exposure to the specific antigen, there's 3 to 6 days after the antigen challenge before things start to happen. This is how long it takes for the microbe, the antigen, to come in contact with the original B lymphocyte that and then have it activate, have it then clone, have the clones change into plasma cells, and the plasma cells start making antibodies. It's quite some time. For some diseases, that's not so bad. Other things, like other antigens, like say snake venom, you don't have 6 days to protect yourself from it. So it's about 10 days when you get that peak plasma antibody, and that's what I said. And that's why most things last about 10 days. And then what happens is the antibody levels decline because we stop putting them out and they're proteins, and so they just end up basically breaking apart. And I mean, we have some of them last, but there's a fairly short half-life. So then the second immune response, the re-exposure, these memory cells respond very quickly because there's so many more of them. It doesn't take any time to meet the correct B lymphocyte. The antibody levels peak far quicker and they're much higher levels because because there's quite a few of them, and these antibodies tend to last a lot longer. They're a little stronger. So again, a very similar graph to the one that we already saw, with the secondary response being higher and faster. Now, there's a concept. You can have humoral immunity by just having the immunoglobulins, by having the specific antibodies. Now, so there's 2 ways of having them. Either you make them yourself, which is called active humoral immunity. Humoral refers to antibody-mediated immunity. It's found in the humors. So the B cells encountered the antigen and then produced the antibody against them. And there's 2 ways that can happen. One is natural, so you get sick. You respond to an infection. You get the bacterial infection, B lymphocytes encounter them, you make antibodies against them, and so you have active immunity because you've made the antibodies, and it's natural because you got sick. Or you can get them artificially. And really what they're doing here is they're taking, instead of a live microbe, they're taking dead microbes, or attenuated, which really means spayed, pathogens that can't reproduce. So they're either dead or they can't reproduce, but their antigens are intact, so they inject them into you, and you have that primary response to something that can't give you the infection. So it's artificial. So it's an artificially acquired active humoral immunity. And so these are called vaccines. And so they stop you from getting sick. But you do have that primary exposure, so that if you encounter the pathogen for real in the environment, your reaction will be a secondary exposure. There's another way of getting them, and that is passive. And in passive humoral immunity, you actually don't make the plasma cells. You don't create the antibodies. What happens is you get antibodies from a different source, from a different organism. So what happens here is that we don't get any memory. The protection ends when the antibodies are gone. So this would be things like your mother gets exposed to pathogens in the environment before you're born. And she makes antibodies to them. Now, her plasma cells don't cross the placenta, but the antibodies, which are extracellular proteins, do. And so when you're born, you have immunity against the pathogens that are in the environment that you're born into. It's probably a good thing. It gives you a chance to survive those first few days of life, maybe first few months. And there's a certain number of these antibodies also pass into the baby through mother's milk, through breastfeeding. It's one of the arguments that breastfeeding is superior to bottle feeding. And that's a natural thing. It's been happening time immemorial. Whatever the mother is exposed to, she makes antibodies against that pathogen, and then those antibodies get passed to her babies, who have some protection from it, but no memory to it. So when they get exposed, they have that protection, or if they're to get exposed. And in a way, it means that they maybe will survive to have a second exposure, in which case then they resort to an active humoral immunity, a naturally acquired active humoral immunity. Now, the artificially acquired is when the doctor gives you an injection. And these are shots. These are injections of serum. They tend to be called gamma globulins. And this is when we have antibodies that are collected from a different organism by drawing the blood and centrifuging and taking the antibodies out. So this would be things like tetanus shots. What they do is they give tetanus, and they infect tetanus into the horses or cows or something like that. That animal makes antibodies against the tetanus, and then we harvest blood from that animal, and then we take the antibodies out of that animal's blood against the tetanus, and we make a serum of it, and we inject that serum, and it gives us the antibodies we need for protection against that infection. It's an artificial acquired, passive. Another example of this is snake venom. The reason that they milk snakes to get the venom is so that the antigens of that venom they can give to large animals in doses small enough to not kill them, but large enough that they have an immune response. They undergo the immune response, make antibodies against that snake venom. We harvest the blood, make the serum from that blood against that anti-venom serum, and then have that on hand so that if somebody gets bit by that type of snake, we can inject antibodies into them to fight that antigen that's in the snake venom and potentially save them. We tend to have these things on hand in hospitals for the local snakes. I doubt if you could go to RVH right now and get anti-cobra venom, because we don't have a lot of cobras, but they probably have a whole raft of Massasauga rattler venom serums. So there's basically then 4 ways to get humoral immunity, to get antibody-mediated immunity. You can have naturally acquired or artificially acquired immunity. You can have naturally acquired active, which is you get infected with the pathogen. You get sick. You have naturally acquired passive, which is you get the antibodies from your mother through the placenta or the breast milk to things that she has been exposed to and is having a secondary response to. Or you can have artificially acquired, which means that you're getting an injection from a health professional. And active, it's called a vaccine, and that's a dead or an attenuated pathogen is injected in you. You have the primary response against it, which means that if you ever get exposed to the pathogen for real, it will be a secondary response. Or you can have passive, which is you get injection of the gamma globulin, of immunoglobulins in a serum form against that specific antigen that you are targeting. So things like tetanus or snake venom, or they used to do it for hepatitis. Now there's a hepatitis vaccine for two of the hepatitis, but they still give gamma globulin for the rest of them. Like it's a good idea if you're going to a place that is known to have certain pathogens, that if you can't be vaccinated against it, then to have an injection of the serum is probably the next best thing. So this is the immunoglobulins, the antibodies. These are in the blood proteins. They are soluble. They bind specifically with an antigen. 5 classes: IgD, M, G, A, and E. I tend to make it M, A, D, G, E. MAG are the 5 classes. IgDs are the receptors on the B cells, and they're used in B cell activation. IgMs have 5 things joined together, and they're released by plasma cells, and they basically, because they're pentameters, they are involved with agglutination. We see the serums that they use for blood typing are IgMs. IgGs is the most common one, and it's really in the primary and secondary responses. It's the one that crosses the placenta. It gives you passive immunity. It's the one that neutralizes, activates complement, immobilizes, does that sort of thing. IgAs really are not that common. They tend to have 2 heads. They're dimers, and where we find them is on your epithelium, and basically their job is to handcuff any pathogen that's trying to get into you at the skin and make it more difficult for them to enter. So they protect as part of the first line of defense. And then IgEs are cells, sorry, not cells, they're antibodies that really bind to the mast cells and to the basophils, and they cause that release of histamine that starts the inflammatory response. So really the B lymphocytes start making some IgGs and IgEs and that kind of thing, and the IgEs really are involved with inflammation. I like this little chart that shows what they do. The antigen reacts with the antibody as what's called an antigen-antibody complex, right? And so what happens? This complex inactivates the antigen by neutralization, by masking the dangerous parts, agglutination by sticking them together, precipitation by making them not be soluble anymore, right? And those 3 things all enhance phagocytosis, because it means that the phagocytes can't get away from, and they eat. So neutralization and immobilization happen here. They also activate complement, and activating complement promotes cellular lysis of the microbe, enhances inflammation, causes chemotaxis and histamine release, and gets inflammation going, and enhanced phagocytosis because it gives the phagocytes something to grab hold of. Nice little chart for the visual learners of what antibodies do. Neutralize and mobilize, agglutinate, precipitate, activate, complement. All of these enhance phagocytosis, cause inflammation, and cellular lysis. Complement does three things. Remember the rule about multiple choice questions and things that do three things. We use the humoral response because it's quick, it's simple, there's a huge amount of antibodies can be made at fairly cheaply. They interact in the extracellular fluid, so tissues don't get damaged for the most part. We can secrete them to the outside of our body through sweat, through sebum, through saliva, through tears. We can put them in the tissue fluid, in the blood, in the lymph. They can go everywhere inside and outside of our body and give us a huge amount of protection. The cellular response are really targeted cells. They get rid of cells infected with viruses or bacteria or parasites that go inside the cells. They help deal with cancer cells or abnormal cells, cells that could become cancerous. They are also involved with transplanted tissue, and that's not a good thing, but it happens. So if you get a kidney transplant, the T cells will recognize there's something wrong and probably attack that organ. This chart is actually a great little chart that shows how all of this interacts, both the specific and the non-specific, the humoral, antibody mediated, and the cell mediated immunities. So basically you get an antigen. The antigen, the microbe, triggers these innate defenses, the non-specific defenses. So that would be things like fever and inflammation and making complement and interferons and all of that stuff. The antigen, it gets eaten by a macrophage, part of the innate defenses. The macrophage becomes an antigen presenting cell. The antigen presenting cell presents the antigen, the processed antigen, to a helper T cell. And it co-stimulates because it's an antigen presenting cell. So the helper T cell activates, clones, has memory helper T cells, and activated helper T cells. The engulfed pathogen, or the antigen, can end up infecting a cell. The cell processes and displays in its MHC1s, which then will activate a killer T cell. The killer T cell needs to be co-stimulated by the helper T, and then it clones. You have memory cells and active cells. The active cells trigger the attack on the antigen. It attacks here, gets them out and gets them eaten. The helper T cells will also activate the killer Ts and cause more macrophages to become more active. And together, they attack the pathogen. The free antigen directly activate a B cell, which can act as a presenter to the T cell, the helper T, or the T cell will co-stimulate and make this be stronger. Some of the B cells that are activated will clone and turn into plasma cells, which make antibodies, which precipitate, neutralize, agglutinate, immobilize, enhanced phagocytosis, activates complement over here to help get rid of the antigen. Some of them become memory cells. So once you've been invaded by an antigen, you start this whole cascade of all of this stuff, all going back to getting rid of the antigen. The whole point of the immune system is to get rid and eliminate the antigen. And while you're at it, you've made memory cells to the B, you've made memory cells to the helper Ts, and you've made memory cells to the killer Ts, so that your secondary responses are much, much stronger, and you will probably not get infected.